is Alina, and I study cell biology. Just as our bodies rely on a skeleton, every living cell needs a framework too. It's called cytoskeleton. This cytoskeleton is made up of different elements, one of them being microtubules. Microtubules give the cell its shape, help it to move, and they are essential for dividing cell into two daughter cells. Microtubules look like tiny tubes organized in a network. They are made up of smaller building blocks called tubulins. Cells carefully control quantity of these tubulins, so there is not too much or too little of them, just the right amount needed. A disruption of this balance, specifically an excess of tubulins, is linked to serious problems like cancer. In my thesis, I study a mechanism that controls tubulin quantity. Cells produce tubulins from instructions of mRNA molecules. When the cell needs more tubulins, production continues. And when it has enough, mRNA instructions are destroyed and production stops completely. This system is very precise, but here is the puzzling part. We don't know how cells monitor these tubulins. How do they sense how many tubulins are there? Just imagine a factory with no quantity control. It could produce way too much of a product. My research tackles this very question. What is the tubulin monitor? How does the cell know when to produce tubulins and when to stop? To find this unknown sensor, I'm modifying tubulin in a way that I can identify which partners it interacts with very closely, just like a painter who paints everything nearby. Then I collect those painted partners and analyze them by the technique called mass spectrometry. By doing so, I hope to identify the tubulin sensor itself, as well as other players involved in tubulin monitoring. Understanding this will be a huge step forward. For example, cancer cells produce much more tubulins than normal cells, and this allows them to divide and grow so fast. If we learn how they trick tubulin control system, we could develop new ways to stop uncontrolled cancer growth with novel or improved treatment options.